students. Think.
Can I start the welcome? Can I start the welcome?
Yes, please go ahead.
Yeah, thank you. Andu sir?
Please, sir.
Yeah, thank you. அனைவருக்கும் இனிய மாலை வணக்கம். First, I put forth my sincere thanks to our management of KALASALINGAM group of institution for accepting wholeheartedly to conduct this 49th International Virtual Learning Series with the blessing of Almighty, our founder, Devathiriti KALASALINGAM Ayya, and with the support of our chairman, Ilayavallal, Dr. S. Sridharan sir, for the 49th International Virtual Learning Series in association with the Indian Pharmaceutical Association, South Tamil Nadu local branch, head office at ARULMIGU, KALASALINGAM College of Pharmacy. Can you hear?
Yes.
Thank you, thank you, thank you. I wish to extend my deep appreciation, and we are proud to have an eminent personality, notorious scientist, well-known administrator, and honorable resource person, Professor Dr. David F L Jenkins, Department of Nanomaterials and Device, University of Plymouth, United Kingdom, to grace this occasion. We thank you for kindly being the valuable part of this session, sir. It is a great honor to have with us for this occasion. Let me acquaint you with the sterling quality of eminent personality who is a brilliant researcher and veteran teacher. Sir, your prestigious presence is a great honor for us and great motivation and inspiration for our entire student community. Sir, we are fully appreciate you for finding the time to be with us in spite of your busy schedule, sir. Most welcome, Dr. David, Professor Dr. David, sir. Most welcome, sir.
Thank you.
Thank you, sir. Next, I welcome all the professor, faculty members, and my dear enthusiastic students for today's session. I hope this session will be a good platform for the students to get a good knowledge. All the students will utilize this great opportunity, explore themselves. I trust that every students will put their effort to uplifting the successful career. All the very best for all the students. Once again, I welcome you all. Thank you. Welcome, Dr. David, sir. Professor Dr. David, sir. Welcome, sir. Please proceed, sir.
Okay, thank you. I say, if you was here, I'd say good morning, but I'd say it's good afternoon. I think it's about 3 p.m. over in Tamil Nadu. There seems a long time now since I've been to India because of all these problems. But hopefully that will change. But I know things are not so good in parts of India at the minute. So let's have a look. So hopefully we can forget about that for now and have a look at something a little different. So this talk is it's really based around sort of the kind of research concepts as much as the actual topics, just to kind of show sort of students who are setting out, maybe beginning their PhD or thinking about it, how some sort of good practice to develop your kind of research kind of processes. So this is a talk. You can see quite a few sort of co-authors, both in the UK and you can see also in India, from IISc Bangalore. They're really frustrated. I think Bangalore is in, I think maybe they come out of lockdown today, I'm not sure. Okay, so we're going to look at two kinds of things. some sensors that we've been working on for detection of neurodegenerative disease, and the platform is quite generic, and so we've been working with Dr. Vikram and Professor Sai Siva Gorti at IRC on applying these for waterborne contaminants, and we'll see we can also preventing contaminants means not only are they there, but can we remove them as well? So it's quite a few things we'll cover. So we'll kind of crack on. I never know what people know about some of these kind of diseases. I guess brain tumors, everybody has heard of. Alzheimer's disease is definitely a disease that's less common in India, but it's definitely where you have a young population. But in the West, with an aging population, Alzheimer's is one of a few kind of these neurodegenerative diseases which cause real problems. Now you may say, who do they cause the problems for? Okay, we'll go on to have a look at that in a little while. So who is the kind of the patient in this particular kind of area? We'll look at, but essentially because we're looking at this with students and research development in mind, we kind of go through a systematic kind of process of developing and validating the sort of sensors to work in these particular areas. We'll then go on and see how we can then apply the similar kind of work that we carry out in this Alzheimer's work on the gliomas. So this is we'll look at what this is really about later, but essentially we're trying to find out, can we determine if someone has the onset of a tumor in the brain without the current need for invasive surgery. And invasive surgery means that it's already advanced quite significantly. The idea is, like when the snowball rolls down the hill, it's much easier to stop it at the beginning than when it's got a big mass and it's got a lot of momentum. So we're trying to see if we can kind of come up with ways of managing, preventing, etc. Another area that we haven't quite got to do yet, but we've made a good start, is to look at alternative sensor platforms. And for me, this is very interesting because it fits into the work on all of the work in Alzheimer's, on brain tumors. It also works on hopefully in the ideas of a real-time monitoring of heavy metals and pathogens in water. So it's quite an interesting platform that quite a lot of people are beginning to explore today. And then we'll go on to look at what we will call disease prevention, i.e. looking at what's in the water and how can we remove them. And we're not looking at dirty water. We're looking at water that looks clean, looks safe, and mostly it is safe apart from maybe one thing. And that's the thing we need to find out and remove. So let's make a start and look at Alzheimer's disease. Now, I do know people say it could be due to large amounts of turmeric that are consumed, that the Indian diet is been very good at preventing people from developing this disease to the extent that it has in the West. But but the question is, who is the patient in this particular case? So how does it affect the person with Alzheimer's? Well, because it's a progressive neurodegenerative disease, the person may first of all kind of notice that they may forget things. Now, we all forget things, of course, but there becomes a point where it becomes more significant. But these kind of diseases, they also they don't manifest themselves in a big change. They manifest themselves a small change, and then you go back to normal, and then you have a small change, and eventually those small changes become bigger changes until the total situation is the disease itself. So as a person becomes more affected by this, then they start to show more significant signs, because it's the brain's ability to control a lot of bodily function starts to degenerate. So people have difficulty, say, with swallowing, some areas of speech. Then it starts to affect, say, their cognitive behavior. And then it gets into the difficult situations where a couple have been married for 40 years. The husband, for example, has Alzheimer's. His wife says, "Good morning," and he says, "Who are you? I don't know you." So, and then they become the challenges. So who is the patient? The person with the disease, we can only look at from our perspective. But for the person trying to look after them, they now have a person that they've loved all their life, they still love, but they don't recognize them. Their body is much more difficult to look after. And finally, the brain will shut down, and then we have to kind of think, okay, what can we learn? What can we do? So these are definitely difficult diseases to detect. So it's not as simple as it is once the disease is displays symptoms. So the problem is, how can we find if there is something in the blood that says this nasty disease is on its way? So we have, like, one problem is this identifying biomarkers. Now, this is maybe the most challenging part of this research, and this research in Alzheimer's is part of a large project. It's a Marie Curie project with around 14 PhD students around. They're based in Europe, but they come from all over the world. In Plymouth, we have Jagriti Sethi. She comes from Delhi. She did her MTech at Amity Noida, which I managed to visit, and she certainly left an impression with them. So people from everywhere. So we're trying to identify the suitable sensor technology. This is, again, it's a challenging thing, because we're moving into areas where the materials become very interesting, but they come with so many challenges. So we have to kind of explore a number of sensor platforms, develop the technology, and then apply it to the biomarkers. And hopefully we can get to a point where people will be able to have this kind of lab on a chip that people talk so much about, and it has been developed in many cases, but we're now looking to expand the platform to this kind of exciting area, because as I said, it leads on to, like, who is the patient, and as you can see, we can identify these, detect biomarkers. Then the big thing, it puts a big burden on healthcare, because this is can be a massive part. The person looking after the person no longer can work; they become the carer. So life becomes very, very challenging. So, as I said, there's been a big part of this project. As you can see, there's been exploring massive data sets using different sort of ideas, data mining, deep learning, the AI, trying to identify sort of markers that we can explore. And some have arisen, and some are already around, which are useful for us to develop the sensors around. But we're not going to be focusing on develop the detection, the identification of the biomarkers, but the way that we can actually develop a platform in a nice logical way. So we started off with essentially a relatively simple platform, and this uses a commercially available sensor, which we're going to be we need to actually make these ourselves now. We're using different materials and create different devices, but again we're completely locked out of our lab at the minute, so we can't progress in this, but this is the kind of sensor that's being used, and it's based around electrochemical sensing. So this was the first sensor that we kind of got to play with here, and as you can see, the idea is to understand this kind of relationship between, say, the current and the voltage under different configurations. And the idea here is to just try and see what parameters will change the response of this system. So we've often used in the electrodes either graphene oxide, reduced graphene oxide, or even graphene. So I hope everyone is familiar with graphene, this nice hexagonal close packed structure, a monolayer. It can be several layers, which are layers on top of each other, not just a thick film. Then we have, for those who are not so familiar, graphene oxide. Essentially, it looks just like, if you had a monolayer of graphene oxide, it looks just like graphene, but it has functional groups attached. So you get epoxy, hydroxyl, and carboxyl groups. And we can see later how that if we actually apply a reduction process, which can be electrochemical or photothermal, then we can actually liberate many of these functional groups, and we end up with a material that has some groups and maybe some defects in the structure, and this becomes known as RGO. But in this case here, we're just starting here with GO. Remember, GO is not a conductor, whereas RGO and graphene are excellent conductors. So this one here is, you can see here how this has been characterized with different concentrations of graphene oxide in the electrodes. And here you can see we develop a systematic approach, and you can see this is the kind of basis. So what happens the next stage, we then move on to further explore the parameters which will change the response that we get from this cyclic voltammetry analysis. We'll see that there are other ways that we can operate these sensors, but in this particular case, it's just current versus voltage in this simple form. And you can see here that on the left, we've shown how the response changes with according to 4 different electrode materials: RGO, graphene, RGO, graphene, and then GO graphene. So again, it's nice and systematic, and we can see how the RGO graphene is the most sensitive surface in this particular case. Okay, so like in many sensor situations, how you operate them also has an impact. And in this case here, you can see how we can change the response by changing the scan rate. Remember, in all these, you can't just keep on increasing and increasing and increasing. So we can see that we've increased here, but if we do keep on increasing, we'll find that we'll start to lose. You can never keep winning and winning and winning. So we've kind of seen how we can develop these sensors, and then we need to try and kind of apply them to a particular situation. So here we've now using the same kind of sensors, but we're using a slightly different configuration. So it's still electrochemical, but we're now using differential pulse photometry. And you can see here that in the detection results that this particular device structure and now has a detection limit of about 1.6 times 10 to the minus 13 molar, which is to a degree better than all of the detection limits which have been reported elsewhere. So this gives us a lot of confidence. So now we can apply this more directly in the area of Alzheimer's. So clustering has been identified as a useful biomarker in the development of Alzheimer's disease. And you can see here that we were interested in two particular issues here. One is the sensitivity and also the linearity. So how does these kinds of sensors detect clustering? We'll look at this a little bit more deeply later on, but essentially we have to form on the surface this kind of antibody antigen sensor configuration, as you can see here. And with this particular sensor, we've been able to go down to around 1 picogram per milliliter with a good linear range of 1 to 100 picograms per milliliter. So bear in mind this kind of number of 1 picogram per milliliter, although we will find it in some of the other research that has been reported. We change to a different kind of units, but when we look on SIRS later on, this 1 picogram per milliliter will be quite an important consideration. Okay, so cyclic voltammetry is a useful tool, and we're still working on in this particular area. So we'll see some of the results that Jagriti has recently published, but some of it's based around someone who was one of the postdocs working within our group, and now a couple of PhD students. They're trying to do the similar kind of detection using the graphene field effect transistor, or GFET. And again, there are some similarities with the work that we just looked at, but this case here, now it's the same process of, can we develop this sensor platform using say known sort of biomarkers. So in this case, we're not looking at a biomarker for Alzheimer's, but we're looking to detect HCG, and you can see here it's a similar kind of configuration where we essentially form our detection channel, which is this graphene layer, and essentially we have to modify it such that we can detect changes in the say the electrical characteristics of this GFET according to this HCG binding event. And so we need to develop the GFET technology. So this became much more of a challenge compared to the electrochemical. We were at the time we would buy in from drop sense sensors, and we could choose different electrode configurations. We can also modify them. But when it came to the GFETs, then this all had to be done within our clean room. So I'm not sure how much people know about graphene. Everyone has probably heard of it, but it's a very, very difficult material to work with. To give you a clue, as to the difficulty of working with graphene. Professor Ferrari at Cambridge and Cambridge Graphene Centre, he kind of has an idea that from the early days of graphene in 2004, it could be 40 years before we work with graphene in the same way that we work with silicon. And that, actually, silicon now, you would say it's a commercial technology. People may micro machine silicon, but people buy silicon wafers. No, universities don't make them. And similarly with graphene, it has become largely a commercial process. It's a difficult material to make, and at the minute it can only be made grown on copper. And as you can see, this is the kind of the starting point. And if you want to get if you wish to get graphene onto your silicon dioxide substrate, then we have to go through a number of processes. So the first one would be to coat with a polymer, PMMA. And then we have to say it's a sacrificial process where we have to essentially lose the bottom layer of the graphene. You have to lose the copper, and then we have to then remove the PMMA. And as you can see from these kind of processes, we end up in the point where we're at E, where we have our kind of graphene monolayer with the PMMA polymer on top. And the normal process has been to use acetone as a solvent. The graphene bonds well to the silicon, so the idea is you wash the PMMA with acetone, but unfortunately that doesn't actually work well enough. It doesn't remove everything, and so we end up with problems when we look at the electrical characteristics of the structure. And so the technique that we developed was to expose the PMMA to 254 nanometers deep UVC. And again, you can't do this for 1 minute, 20 minutes, or 3 hours. You have to find the optimum time, and we found that around 20 minutes exposure to this wavelength was able to allow the dangly bonds to kind of weaken. And so when we then applied the acetone wash, we found that we pretty well removed all of the PMMA. But for now, don't think that's now solved all the issues. It solved this issue. Okay, so what does this mean? It means that we can kind of again characterize these devices, and you can see the on the left hand side kind of A. So this is just a conventional kind of photoresist lift off process on graphene, and then we have the other process using a deep UV, and you can see now that looking at the Raman signals, not just the fact that but as you can see, the on A, the signal, the Raman signal is quite noisy, and on B, this is not the case. But it's also important to look at the scale to see the intensity, which is a measure of the signal, is the counts. So without deep UV, we go up to around 1,000 counts, whereas we go to 12,000 using the deep UV treatment. We also remember Raman is a point measurement, but if we map it, we can see. So what we're looking for here is to have a fairly homogeneous map of the surface, and that you can see this is what we've been able to achieve. And then we also validate this further using AFM. So you can see there's an optical image on the top, and you can see the line scan of the AFM shows that you can see the actual residue on the surface, which has now been largely removed by the deep UV process. And so we can then go on to apply all of these processes now to create this this channel. So you can see now we're looking at the gate within the device. The source and the drain are formed by evaporating chrome onto the silicon dioxide, and then we sputter gold onto the chrome to create the source and the drain. And the working electrode is the graphene channel, as you can see. And again, we have to go. So how does this this sensor work in sense of how does it perform? As we go through this process of trying to validate it and understand it. And so obviously what happens when you've got nothing, just a bare device. So this gives us a reference response, as you can see. And then on the right, on B, we now are exploring what actually happens under different concentrations. And you can see here for the HCG, as you can see, concentrations are like 1 picogram to 100 nanograms per milliliter. So we're in these devices, we're looking at the resistance of the channel. And so again, you can see on the left hand side, you can see how this sensor evolves as we go through from the start, the base, as we introduce the linker, and then we have the antibody, we have the BSA, which is the blocker to limit any interactions outside of the antibodies. And then you can see finally what happens with the HCG. And again, on the right hand side, you can see that we're down to this sort of one picogram per milliliter, which seems to be where we're kind of plateauing at the minute. So we now have to work out what can we do. And at the minute, I don't have the answers for how do we go beyond this at the minute. But there will be a way, I hope. Okay, so this is all part of a project called BBDAG. And you can see there's Jagriti. She's been working on this now for 2.5 years, and she's sort of currently now trying to look at winding up her research and trying to write up. She's oh, hold on. This is some of she's recently published this this paper on this sort of label-free biosensor based on reduced graphene oxide. And again, you can see, again, it's systematic development of the sensor. In this case here, it was playing around with adding an additional factor. This is going from a macro electrode to a micro electrode. And again, this gave us an improvement from 10 picograms down to this sort of 1 picogram per milliliter for HCG. So sort of, and you can see now some of the cyclic voltammetry results. And finally, we come up with a means of trying to give a kind of degree of a performance measure of this. So, and as you can see, that the results take us down into the sort of very low concentrations, but importantly, we have this nice linear response. and maybe this is the area, one area where maybe now further signal processing may take us a little bit further. That's quite often the case. A physical system can often go an order of magnitude further with suitable sort of software tools applied. So this is where we got to so far in this particular area. So in parallel with this, we have another project started. So this is about a year and a half old now. So we're about halfway through, and again, it's 14 students all around Europe. I know there's definitely one student from India I met in Italy working on this. Mina, who's working on this, she comes from Iran. So she's working really hard and really keen to get back into the lab. So what this is about is essentially these gliomas, a type of brain tumor, and so they're like many tumors. They're aggressive, fast growing, and remember with tumors, you really have to stop them before they have any chance to get going. And at the minute, if you have any symptoms, and typically these could be severe headaches, for example, is one common area where people may show some signs. You then have to have like biopsies and say various, even though they're not invasive physically, they're quite personally invasive to have CT scans, MRI, etc. And so we got a number of projects within this overall project looking at detection mechanisms. Again, we can apply these into all of the other areas that we are working in. So these are the particular cells that we're trying to identify the presence of, and we're trying to identify, well, maybe not the presence of, but the potential presence of them. So are they going to be formed within the structure of a person's brain? Again, the same big problem. Biomarkers, and again, one of the PhD students is based in Plymouth in a medical school, working on in this particular area, to try and identify these either biomarkers. So we're looking at biomarkers. In this particular case, Mina is actually looking for something slightly different. So rather than looking for this this particular biomarker in the way that we had looked at before, Mina is trying to identify DNA methylation, and for this particular case, within the project itself, there's 3 other techniques which people have been using, but Mina's been working on electrochemical. So there are the GFETs. There's a group in Belgium, I think, working on digital ELISA. Group in Italy are working on plasmonics, and in Plymouth we're working on electrochemical, and similar kind of methods are being applied here. Mina's doesn't want me to share her results just yet because she's just writing a paper. So hopefully that paper will be ready soon for submission, and then we can tell you a little bit more about what she's found. But again, it's using this. It's a very similar, because these are kind of generic sensors. But the interesting one for me at the minute is this kind of surface enhanced Raman spectroscopy. It's become a tool that's quite fascinated me, not only in these particular bio areas, but I've also developed a little bit of an interest in nanoplastics as opposed to microplastics. So for all of you sort of biologists and sort of bio-aware people, nanoplastics are are very interesting to detect, because these actually are disruptive on the cellular level. And so unlike microplastics, where we don't totally know the actual outcome of microplastics within aquatic species, and then ultimately with man or woman, we do know that nanoplastics and nanoparticles have to be treated with great care. And so and SIRS is maybe the best technique to detect them. So why is SIRS so important? It's one reason is because the Raman signal is very weak, as you saw with our earlier data. When the sample wasn't very good, the signal was very weak. It wasn't such a useful way to work. This is because the Raman signal, which is either classified as a Stokes or anti-Stokes scattering, is something like 10 to the 6 times less than the elastic signal that would dominate. And obviously this is filtered out. But with SIRS, we take advantage of plasmonics to enhance, because this is about the electric field that interacts with the vibrating molecules. With plasmonics, we're able to actually enhance the electric field due to something like evanescent waves, which happen when light is incident at a certain angle. And with spherical objects, then we can always satisfy that criteria. And if we get the particles, so to form the surface, we'll see we have say particles on the surface, and if they get quite close together, then we can get these kind of hot spots where we can even get a greater enhancement, and we can also get some electrochemical enhancement. So, and the good thing is we can use a SIRS chip in a conventional Raman system. And so you may think, okay, that's quite easy, but these devices are hard to make. And so this is work I've started with Bing Lee. He did his PhD at Plymouth. He's now a research fellow at Imperial in working on various kind of medical and biomedical applications. And so we're trying to fabricate these devices, and I do have 2 of these in my drawer in my office, but I can't do anything with them at the minute. So the idea is to kind of, again, it's sort of we have to use sort of various kind of electron beam lithography to form these kinds of structures, and then we have to coat them with gold. So essentially we're getting these little gold spheres on the surface, and with them being close together, we can get significant enhancement. But what's so great for me about these is that, for example, with DNA methylation, we can have samples of methylated and unmethylated or non-methylated and detect them at the same time. So this has a lot of implications for the work that Mina's been doing, but it also has lots of potential for the work that we're doing in Bangalore on water quality, where it looks as though we could develop a portable tool or a system that can be plumbed into for real-time monitoring. When binding events occur, we'll see changes in the Raman signal, but we can also then flush the surface with a change of pH in water, and we can then keep using this sensor chip continuously, maybe not forever, but for a long time. So this kind of takes us on to our last, just been invaded by my pussycat. So this takes us on to this kind of area we call disease prevention. Again, this has become a massive part of my research and personal life, I think, as I got into, say, water quality. So I'm quite heavily involved in the India UK Water Centre. I'll send a link later. We have a webinar. I think it's the 4th of August, if I'm correct, and we're trying to look at the issues of water within the COVID-19 pandemic. And I know that I've been looking at this recently in rural India, where the water issues and COVID-19 and how we kind of manage it have become quite a integrated issue. So to give you, so why is this? So what do we see here? So on the left, we can see, I'd say this is a lovely chalk stream in lovely southern England, and it ticks every box that you require in terms of water and the environment. So that river is full of fish. The water will be clear. It will be low on contaminants. What we also know is that all the vegetation around the river, within the river, are helping to clean the environment, and the fact is that just looks healthy. Whereas in Tamil Nadu, in Chennai, you can see this one here. This is in Tinagar, which I took a couple of years ago. Well, I'll let you comment on that. I won't, but you can see it's not quite right, is it? And this is a river possibly was a source of drinking water 50 years ago. So we want to go from the right to the left, and we do know that there are within the rivers lots of problems, and yes, there are water treatment plants in India, but not like we have in the UK, and certainly not as many. In the UK, we could be global leaders in this particular area. So what we're looking at is what people do within their homes, within industry, etc. This wastewater. Now, sadly, in India, if this was industry, it just goes straight into the river, and it shouldn't. But people are hiding pipes and pumping it into the Ganges. I've seen all of this from LiDAR from IIT Kanpur, and it's absolutely atrocious. But often in domestic situations, this will go straight back into the river. Okay, but what we want is a situation like we have in the UK. The microplastics come into this area. We're trying to sort this one out, but you can see here there's it's a fairly sophisticated process, and this is only to turn water into a safe form to go into rivers, seas, to be a source of surface water and groundwater. Now, if I go to anyone's home in England and they say, would you like a glass of water? I might say yes or no, depending if I'm thirsty or not. Whereas I know in India, I have to ask about its source, because I know that, for example, this particular processing is not happening. So the water that actually goes into here is already pretty good, but it then goes through quite a sophisticated process system, as you can see, that yields water which is just totally safe to drink. It does produce some areas of I wouldn't say concern, but it certainly does raise some questions about the sludge. The sludge is used for fertilizers, and people sometimes comment on it a little bit smelly, but it should be safe in terms of the environment. It also is a means of producing methane as well, can also produce. So that's the situation we've got, and we're kind of interested in, again, the same kinds of sensors, but developing a different kind of strategy. So we know metals are often geographical or industrial in nature. So in the UK, we have the same. In southwest England, we've had water that would be contaminated from arsenic from mining. But I know in India there are problems of say copper, cadmium, lead, nickel, chromium, etc., etc. Some of them are geographical. Some of them, I know in Chennai, chromium from leather industries is a problem, and also the pathogens. And of course, now we have no idea where SARS-CoV-2 comes into this. That's the novel coronavirus. We do not. This is totally unknown as to whether this is something we have to consider here. But we do know that this causes a lot of infant mortality. I know things are getting better, but there's still a long way to go. So we're interested to come up with this kind of again, all low cost. So developing a kind of say GFET devices. And although we say they're using graphene, it will be related materials, because the problem with graphene is that it oxidizes very quickly. And so we're looking to develop not only hermetically sealed systems, but ones using maybe RGO or maybe some other 2D materials that have a kind of a longevity. Whereas the lifetime of a exposed graphene GFET is about 20 minutes before you have to think about what to do next. So we're trying to develop an array of these sensors to detect maybe 6 pathogens. Now, I should have updated this slide because we are no longer working with Next Big Lab. We're actually doing all of this in Bangalore, where we will be once we're back in again. So in Bangalore, we've already working with Smile, the spin-out company of Siva Gorty, and so we developed our own 3D bioprinter, and we're looking to kind of come up with a system again so that's a detection system, and then the group there has a lot of experience and published a lot in this area of microfluidic devices. But ultimately, what we want to try and do is to come up with something like this, where we can have our sort of sensor to detect contaminants. But the key here is, okay, so these contaminants, especially if they're pathogens, are definitely migratory and transient. And we'd like to be able to use a phone and say, so if you go to a village, there could be a bore well, you pump the water, and every week or every month, we measure the water, what's in it, send it to a server, and we want to produce a map of these contaminants to try and understand what's going on. And this will be a totally free open source website that anyone can use globally to see what's going on. Okay, moving sort of swiftly onto the kind of the last part of this. I hope I haven't gone too long, but I do talk a lot sometimes. How can we remove these contaminants? So we've the work in Bangalore with metals focused on lead. We're now working on E. coli. But the work on filtration has also been around lead. And the idea was initially to develop membranes. Group in Manchester has tried, lots of groups tried. But we could never get our head around the fact that we had a limited kind of volume that we could work with in that kind of configuration. So Jonathan Bloor is, again, he's starting to write up his PhD at the minute. He developed these very user-friendly materials, so they're biocompatible, biodegradable. They're made from graphene oxide and seaweed. Essentially, they're all space. So within a small volume, we have an enormous surface in which we can actually adsorb contaminants the process is charge. So the pores are very large compared to what's passing through. But the idea is as the water with, for example, lead passes through, the lead gets adsorbed onto the surface throughout the structure. It's quite these materials are quite adaptable, and again, this is some work that we've kind of been doing with Bangalore as well, is developing, playing around with these, so we can make these gels to be hydrophilic, hydrophobic, oleophilic and oleophobic. So lots of potential here for the work on nanoplastic filtration. But if I can play this video, you'll see, I hope you're going to play. So you can see here we've got this dyed rapeseed oil mixed with water, and an aerogel is just between two tubes, and as it passes, so now we pour it in, and what we'll see is that mixture is not only separated in the sense that oil and water often does separate, but in this case here, it's separated via this, only the water is allowed to pass through. So the aerogel is then hydrophilic. So it doesn't like the oil, but it likes so the water will go through and the oil will stay. I'm not sure if the water, if Johnny ever drunk this water, but it should be safe anyway. It's only rapeseed oil with a small amount of safe dye. So we can sort of move on to the kind of the next. Okay, so another issue in the water that is that could be available for consumption is obviously dyes as well. So we can also use these gels to remove methylene blue in this case. And again, we can see we can remove these this dye quite well. And if we know that if we put an aerogel into a solution with methylene blue, ultimately you'll see that gel will be full of dye and the water will be totally clear. So then we go on to, again, it's systematic development. Again, lockdown has put us in a position where we can't take this to the next stage. So this is looking at lead in water. So again, Johnny's played around with very, very high concentrations, way above the kind of requirements for drinking water. But we wanted to see how much it could remove, what the capacity was. And they also know that we can also regenerate them. And you can see here, so this is a long time. This is 2 hours. But you have to look at this area here. So this is the area where we're really interested. What happens in moments, in a very short period of time with a very low concentration. So the idea is a gel like this could go into either within a pump system or even in bottles. So you have it a little filter in a cap. So when you drink, the water will just pass through the aerogel, removing the contaminants. And again, it shows a very, very high absorption capacity. So these filters can work a long, long time. Afterwards, they can either be regenerated or incinerated. But they're also the idea is very, very low cost. Okay, and another thing. So all these materials all come onto different kinds of other applications. So what happens if we configure this aerogel such that we can apply a very small electric field, or maybe the electric field may be quite high, but the voltage required is quite low. And we know that with this particular configuration, then we can actually pass brackish water, not seawater, but very lightly salinated water into an aerogel, and again we can actually remove the ions, and the water that comes out is then safe for drinking. So low voltage means we can do this with solar. It's much cheaper than reverse osmosis. So lots and lots of kind of potential. And I know there's a group at Amrita and Chennai, which are working on these particular devices as well. And again, we're looking at, for nanoplastic. So okay, just a little summary then. We've hopefully shown a systematic approach to developing electrochemical and GFET sensors. We know that in the bigger picture of this work, the biomarker detection and identification is key, but we do know that so far we can get very high detection sensitivity. So ultimately, with the right biomarkers, a small droplet of blood dropped onto the device allowed to dry will be a point of source diagnostics, and hopefully something which will improve quality of life. That's the main thing. If we can make things better, make a difference, then it's good. We may find that SIRS is another tool, but that will be a more expensive platform, whereas the other sensors are designed around low cost. We can apply our material development to make these aerogels. So we see lots of applications: removal of metals, pathogens, dyes, and for desalination. Now, if you want to know a little bit more about not just the work we do in Plymouth, but the work that my colleagues are doing, there's a recent article. I think it's available in August for so this is a review article that looks at essentially graphene-based systems for the detection of neurodegenerative biomarkers. We have another paper in preparation, and also a book chapter should follow soon. So lots of things which will be available soon to hopefully whet your appetite. But say this is a teamwork. So couldn't really done any of this without a massive team. So you can see, so a lot of my Genwal Pan, Kamal Islam, he was the guy who began the work on the GFETs. Lots of CC colleagues from UK, India, China. It's a big team, and say you can't work on your own these days. You need to work as a team, work globally, work internationally, and hopefully we'll make some progress. So I just finally to say thank you very much. I hope that wasn't too long.
Thank you, Doctor. Thank you, Doctor. Thank you, Dr. David, sir. Thank you, sir.
So I hope that was okay.
Yeah, thank you, Doctor. We had a wonderful and informative presentation, Doctor.
Yeah, so
Professor.
any
questions?
I can share the PowerPoint with you.
Okay. Questions? Any questions?
This is where everyone goes quiet.
Any question? Any question?
If anyone wishes to ask me any questions by email, my email is there. Just contact me. I know often people get shy asking questions, and there's no such thing as a silly question. Okay, if something's not clear, ask. Don't keep wondering. Yeah.
Anbu sir.
Yes.
Hello.
Hello.
Yes, sir.
Is there any chat box question, sir?
Pardon?
Anbu sir.
Sir,
Is there any?
no question, sir.
Okay.
Okay. Okay, Dr. David sir, very kind of your presentation.
Okay. Yeah, thank you.
On behalf of our management of KALASALINGAM College of Pharmacy, hereby express my wholehearted thanks to you and your university for given such an informative and wonderful presentation, Professor Dr. David.
Yeah, so hopefully.
I hope this interaction will motivate the students and encourage.
So hopefully I can visit when
them in active learning, really.
Yeah.
Dr. David sir, Professor Dr. David sir. David sir. In future also, let's have a good collaboration for the development of our students' career, David sir.
Of course, yeah.
Thank you, yeah. Doctor.
Anyone can contact me.
Thank you. Another one more request, Dr. David sir. Another one more request.
Yes.
Kindly refer your colleague or friends for our future webinar, international webinar session.
Okay.
Kindly refer for our future webinar.
Okay. I can ask.
Thank you.
Okay then.
Thank you, Doctor.
Okay, so have a good day. Have a good evening. Right, I'll stop showing my. Let's stop. I don't know.
Yes, sir.
Yeah.
Thank you, sir.
Take care, Dr. Govind.
Okay. Right.
Dr. David sir, thank you.
Okay. Bye.